Hi. Um, all right, looks like we're live. All right, hello everyone, and welcome. I want to say a big welcome to our guest speakers, and I know for some of you it's a bit earlier than it is for us here in the UK and America, so we're especially happy that you could make it and share your experiences with us. Massive thank you also to our co-host DoCSoc for helping the Synthetic Biology Society, SynBIC, make this possible. We will now hear from our speakers about how software is making an impact in biotechnology and what the future is. Without further ado, I would like to introduce our first guest speaker, Dr. Yaoyu Yang. Dr. Yang has done automation and synthetic biology at the University of Washington and is now a principal software engineer at Ginkgo Bioworks. If you'd like to share your screen, we're ready when you are.
So here's the problem. I don't, the share, is there like any other thing I need to click? I
look into it very quickly, but I think you should have a, is there not a button there to share the screen?
So the share screen is disabled for me. I don't know why.
Okay, let me take care of it.
Also, I think I'm still in the backstage.
No, you should be on the stage. I think.
Oh, I'm already on stage. Okay.
Okay, yeah.
Sounds good.
Maybe it'd be worth doing a speaker intro or something. Screen quickly before while I sort this out.
Yeah. Yeah. Would you like to introduce yourself briefly, Dr.
Oh,
Yang?
Yeah. Oh, should I introduce myself or? Sorry.
Yeah, that would be nice.
Okay.
I did a very brief introduction, but
yeah. My name is Yaoyu. I currently works as a software engineer. Before that, before joining Ginkgo, I was at University of Washington. So I started as electrical engineering major, but at my first quarter at UW, I bumped into this email where the professor sending out an advertisement of a course called Introduction to Synthetic Biology. And in that email, he was talking about programming cells to change the future. I was like, holy moly, this could be the future. So I actually took the course and just got really into this business. So I joined his lab. So the professor is Eric Clemens. So I become his PhD student and worked on sort of learning all the sort of things about the molecular biology side of things. And then like doing like molecular cloning for quite a couple of years, building synthetic biology like circuits in cells. So in the meantime, I found this like sort of lab work really sort of tedious and repetitive, right? So we got really interested in how to automate this. And the solution we came up is sort of a low-cost automation where it involves people. So we build the software to sort of building abstractions on top of the lab processes. And that allows us to basically hire undergrads and to train them in 1 to 2 weeks to become really sort of sufficient in like executing the molecular biology protocols. And then we can actually leverage their skills, right, that train via the software to actually instruct them to do more complicated biological protocols. So what ends up is you build this sort of software abstractions that you models your entire protocols and workflows, and you can sort of starting to focus on the design level of how you want to design experiments and not worry about like the experiment execution part. So basically separating all the design and execution. So that was sort of a lot of work I did on the sort of writing that software to implement that. So that sort of leads me naturally into Ginkgo, where Ginkgo is really heavily leveraging software and robotic automation to do cell programming. So I've been in Ginkgo for almost 4 years plus. It's really amazing to see how fast we're growing, how sort of Foundry is almost like tripling the capacity and throughput every year. And the challenges come with them, how the software and robotics actually really helping that scale. So that's sort of my introduction. Did we fix the slide problem, or should I maybe log out and log in again?
Yeah, so we'll just be having just the current speaker on the stage, and then you'll be able to share screen. Yeah, if the other people can hop off. Okie dokie. Yeah, you should be having the option now.
Oh yeah, you're right. So I should open system. So entire screen. I suppose that should work. Can you guys see my screen? Or no?
Not yet. Does it appear? Is it loaded on yours?
Yeah, it says entire screen I can share.
You should probably select something like the screen to share, something like that.
Yeah, it says it wants to share. So give me 1 second. Maybe I can try another browser, see if that works.
No worries at all.
Yeah, 1 second. Okay. For whatever reason, my screen share doesn't work. Maybe you can try other speakers to go first while we figure out the technical problems.
If other speakers are ready.
Yep. Zoey, would you like to go next, as you were up next anyway? Zoey Herm is from Riffyn, and she did her PhD at UC Berkeley and a postdoc at ETH Zurich before starting as a senior scientist, first at Mosaic Materials and now at Riffyn. So we'd like to give you a warm welcome and share screens when you're ready.
Hello, good afternoon, everyone. Let's see. Is my screen sharing?
Yeah,
Yep.
that looks good.
There we go. Okay, great. So thank you very much for the introduction, and thank you for the opportunity to be here today. It's something I've been looking forward to, getting to talk to everybody. So like Olivia said, my name is Zoey Herm. I work at a company called Riffyn, and I'm going to explain what we do throughout the course of this talk and what I do at Riffyn throughout the course of this talk. I titled these slides "Data is the New Science" because, as you'll see kind of throughout the slides, I am trained as a scientist, but now I work in software, and that's because I am very passionate and excited about data in the scientific field. So we're all kind of used, I don't know, at least I am, like when you think about science, you think about somebody in a lab, maybe with a lab coat on and a paper lab notebook where they're taking lots of notes and they're looking at a microscope. I thought that this was kind of a trope of what you think of when you think of quote unquote science. And similarly, like when you zoom in on a lab notebook, you think of something like this. Over the course of my career, I have probably filled up, I think, at least a dozen of these, 120-page notebooks, and they all look exactly like this, writing and, printing out figures and pasting them in, stapling in big packets of, data printouts, images, that kind of thing. But this is obviously not modern, and it's very analog. But modern science looks something a little bit like this generally, where you look at your lab notebooks and they're just kind of something similar to Microsoft Word documents. So we've gone from analog to digital, but we're still, kind of thinking in terms of how you think when you're looking at, digital data or analog data capture for science. So I didn't want to I Googled here generic electronic lab notebook. So hopefully this picture kind of gets the point across that, you have something that's like unstructured data entry. Maybe there's some links to some files somewhere in some kind of a folder or directory structure. So essentially the exact same thing as paper lab notebooks, but just digitized. And that's what like modern science generally looks like. But what modern science actually needs is a lot more complex than that. I highlighted, this laundry list of items that you need if you really want to do science properly and you really want to harness the power of all of the data that's being captured in a, synthetic biology or other lab. You need to be able to do things like link your data across unit ops. You need all of your data to be exportable such that it's ready for any kind of query, ready for any kind of machine learning. And you need it in like a non-proprietary data export format, like a CSV. You need to be able to do structured data capture because you're not going to be able to query things if they're not structured. But you also need to be able to do unstructured data capture because, you go into the lab and you write down if you're thinking about synthetic biology, you write down all of the parameters for your fermenter, you write down all the date, all this kind of important information about the experiments that you're running. But you also need to just sometimes write down like, the temperature controller was out today, but it was really hot in the lab. And that's not something that you can model. That's not something that you can, that you can use in some kind of visualization. But it's information that you do need to capture when you're doing science. And so you need to be able to capture data. You also need to be able to import data from, any litany of scientific instrumentation. So these top 5 pieces are all about data because that's kind of what this talk is about. But when you're thinking about data capture in modern science, there's also, dozens more criteria that you need to meet if you really want to kind of get it right in terms of how you're doing your data capture. So you need to have things like cloud architecture. Oh, sorry about that. Cloud architecture, obviously. Things like audit trailing, security compliance. I won't go through all of these, but you want to be able to send people notifications. You want to be able to do calculations and logic within your data, this kind of thing. And so that's what my company does. That's what Riffyn Nexus does. This figure here, it kind of shows where we like to locate ourselves in the conversation of capturing data in science. So the pictures that I showed you on the previous slides were this electronic laboratory notebook, which are like experiment centric. So you go into the lab, you do an experiment, you write everything down that happened. There's also another way that people capture data in modern laboratories, which are these things called LIMS, Laboratory Information Management Systems. And these are databases, and they're great and they're powerful. But the downside to like a standard database is that it's very rigid, very structured. And so you have to do your experiments every time you do an experiment, you have to do it the exact same way. Otherwise, your database isn't going to be able to handle any kind of changes or permutations that you do in your like actual lab work as time goes on. And so Riffyn, we position ourselves kind of in the middle, where you're not thinking about individual samples, you're not thinking about individual like items that are going on in the lab, and you're not thinking about experiments either. You're thinking about processes, you're thinking about workflows. And so what that looks like is something like this slide. So this is just one screenshot. You're not going to be able to walk away from this talk like, knowing everything, all the ins and outs about the software, and that's not the point. The point is just to show you that Riffyn is really cool for a lot of reasons. I'm very passionate about the company and about the product. What you do when you're entering data in Riffyn, instead of something that looks like just a database entry, or instead of something that looks like a Word document, you kind of have this hybrid approach where you have something that looks like a process flow diagram, and you define all of the steps that go into the experiment or the workflow that you're doing. And then you can define all of the data elements, all of the data objects that are associated with each one of those steps. And you can change it over time. You can incorporate lots of information like your procedural information. You can attach videos in here if you want, et cetera, et cetera. And when you enter your data this way, it just kind of shakes out that it's very, very powerful. You can export all of your data, like I said, for one lab, for multiple labs, connecting information across multiple labs. There's just a lot you can do when you organize your data this way. And so, what it kind of looks like, just kind of giving one more different view into how the software operates. You have your kind of moment where you define what you want your process flow diagram to look like. Then you go into the lab and you capture all of your data live. Either you can do manual data entry, but you can also integrate with things like data historians, Excel, instrument automation, different kind of scripts and that kind of thing. So there's a lot of ways to get data into Riffyn. And then you can collate lots of experiments over time and across processes, like I said. And then once you have all of your experiments exported, then you can analyze it in any kind of external tool. And that's where you get all of your really great data insights. And so, like I said, this is a really powerful approach. This is a really powerful platform. These are just a couple kind of pull quotes from some of our customers who can do their projects twice as fast, or with 25% less, headcount or time, 20 times faster in terms of managing your data, in terms of doing your data cleaning. An Imperial project that I'm going to talk about at the end of this talk, they took something. It took them 9 weeks to develop a tool that was ready to go into the clinic, et cetera. One of my favorite ones when I'm just talking to people casually about what my company does is that people get really excited about is when you are organizing your data really well, when you're managing it very deliberately. In this one specific example, this group was able to minimize the number of animals that they were using for animal testing, because when their data was so structured, they were able to gain more insight from less data, therefore like saving the literal lives of animals. You can also make I know that most people here are on the in silico side, and so you can also make really, really pretty images, pretty visualizations when you have all of your data really cleanly structured and you're spending less time data cleaning and more time doing data analysis. So I just wanted to throw in here just a couple of cool scientific visualizations. This is gene expression. This is some anonymized customer data. This is for antibody bioprocessing. Once again, just using it. The previous one was using JMP as a tool, JMP. This one is using another statistical software package called Spotfire. And here's another one. This is actually anonymized customer data that I collected working with a customer where they were doing 2D mapping. 2D mapping of these samples and these giant instruments to see if there were any kind of edge effects or anything like that. So you can make really pretty visualizations. You can also, I'll talk a little bit about this later, but this is just another pretty picture showing that one of our customers, Novozymes, they were able to capture 25% of North American market for yeast for ethanol manufacturing in just a year and a half. So when you are harnessing the power of data, you can take your R&D a lot farther, a lot faster. And so that segues into why I left the lab. So as Olivia said, I'm trained as a chemist. And so my career up until I moved to Riffyn was really cool and really exciting. I got to do a lot of really fascinating work that I'm very grateful for. And so when I told people that I was leaving the lab to work at a software company, people were like, oh, aren't you going to miss, the excitement of all the things that I got to do? So this is just showing, what I'm showing here is, I don't know how great my career has been up until now. So in 12 years before I went to Riffyn, I discovered brand new molecules that had never been synthesized ever before and characterized them and published them. I published academic papers on hydrogen storage for fuel cells. One of my things that I'm most proud of is this paper about improving efficiency for petroleum refining using materials chemistry, which doesn't sound that cool, but when you think about the fact that petroleum refining is 10% of global energy usage, you make that more efficient, that's packs a big punch. I also got to work on decontaminating soil. I got to travel to India to take soil samples and then take them back to Switzerland to do analyses on them. And then I worked at 2 startups in the Bay Area as well, making palm oil from natural gas to save the environment. And then I also was the first full-time employee of a carbon capture startup. So I got to build that for a few years. So this is really cool stuff. But when you compare that to what I've gotten to do at Riffyn in just not even quite 18 months yet, I've worked on a team that in such a short amount of time, we analyzed fermentation data for cannabinoids and other industrial enzymes. We got to do both R&D and manufacturing data for these proteins that are just notoriously complex, which is something that you very rarely get to do when you're actually doing lab science, is do both early development and the actual commercialization. So that one was pretty cool. Lots of sustainable biotech. So, when you're thinking about sustainable biotech and you're thinking about building a platform, you don't have to kind of focus on just one product. You can do a long list of products like pharmaceuticals, cosmetics, fuels. We get to work on alternative meats. I get to work on this one is just kind of fun. A company that is improving the enzymes that go into bread making. So I got to play around with that data a little bit. Lots of large-scale agricultural data, like that green farm slide that I showed before. I get to work with a lot of agriculture customers, and countless pharmaceutical projects that we get to work on, and lots more. So that's what I do at Riffyn, is I work on this team, and we work, we're all scientists on our team, and we work with scientists at customers of Riffyn, and we kind of help them get up and going, get all of their data into our platform so that they can use it to make all this cool progress in R&D. And I'm not the only one, so I just thought that this was a, fun little segue. So I was thinking am I, among kind of my best friends from grad school, am I the only one who is, am I unique in the fact that I left the lab and am working in more software computational field? And no, so I audited my 5 best friends, the ones that I still talk to, every week or so from grad school. So your friends in school now are, many of them might be friends for life. I went to UC Berkeley for my PhD in chemistry, and depending on who you ask, it is the best or the second best or the third best chemistry school in the world. This is just the US News thing. And so I'm not trying to brag here. I'm just trying to say that everyone I know from grad school, they're obviously good at applying for stuff. If they can get into competitive programs, they could have a lot of career options. And 5 out of 6 of us are not in the lab and are doing, one person's a lawyer, but the rest of us are all doing computational or software work in science adjacent fields, except the one who is still a holdout, and he's doing really cool stuff, making electrochromic windows in the lab. Okay, so with that, so I've kind of talked about what my company does. I'm doing okay in time. Talked about what my company does. I talked about where I fit into that, why I think data and science is so compelling as a career. and so powerful in terms of what you can get done in however many years you have in your career. Now I'll just talk about one specific example of something that I've gotten to work on at Riffyn, and it is a good example because it's something that I did collaboratively with Imperial College, and this is about pooling COVID tests. So this is obviously very relevant to everybody on this call. So essentially, normally when you think of getting a COVID test, you get your swab, and then that swab gets put into a tube, and that tube, gets manipulated a little bit and then put into a machine, and you get your positive, negative COVID result. Couple problems with that. One is that one of these tests, the qPCR, you've probably heard that term like in the news. That's what most COVID tests are. qPCR tests. So one qPCR test is very expensive. And especially if you're doing screening testing, like an entire hospital or an entire school, that cost really adds up to astronomical amounts of money. So it's very expensive, but also something that I didn't know really until I didn't know the magnitude of it until I started this project, is that labs, like COVID testing labs, are still fighting over supplies to get these tests done. So even if they had infinite funds, they would still be limited because the pandemic is global. So things that you need, like sample plates, the reagents, like I said, you have to do some manipulations. You can't just take a COVID swab and put that into a machine. You have to do some chemistry on it first. They're fighting over these things, and they're really, really limited in what they can get. And so there's something you can do called pooling, and I'll explain what that is. So first, I just need to explain what, if you haven't ever interfaced with this, what a plate is, what a sample plate is. So these are ubiquitous in scientific laboratories, these things that look like, I don't know, they're maybe a little bit bigger than an index card. And they have these little wells in them. They're called, if there are 96 of them, which is the most common, they're called 96 well plates. And you can take little bits of liquid or suspensions or solutions and put them in these 96 well plates. And then those are what you put in the qPCR machine. So you need to kind of have that in your back pocket to understand the rest of how pooling works. So what this diagram is just showing is that you have your swab, it's in a tube, and then you take those tubes and you array those into these 96 well plates. And then how pooling works is, so let's say that you have 3 of these 96 well plates, but you can take advantage of the fact that most people's COVID tests come back negative, especially when you're doing like large scale screening, like an entire school. And so if you were to take 3 of those plates that I showed on the previous slide and take a little bit from the top left from each 1, 2, 3, and put all of those into the top left of this new pooled plate, and you put that in the qPCR machine, now you're going to get this one kind of positive result from that one well. And you will have gotten to do many fewer tests for essentially kind of like the same information. Here in this case, let's see, what is it? There's 12, just because it's a cartoon, there's just 12 here per plate. So in this scenario, you're doing 36 tests. And in this scenario, you're only doing 12 tests, and you're getting approximately the same information. And so how that works moving forward is you pool those plates, you run your qPCR test, you get your information that, okay, one of those wells has some COVID in it. And then what you have to do is you have to go back, you say, okay, that top left well, that came from these 3 plates and these 3 different wells in those 3 plates. So you have to deconvolute it, and then you have to make a new plate for those and then see which one of those 3 was the actual positive one. So that's how pooling works. And the reason that we got involved, that Riffyn got involved, is that deconvolution and then rearraying of positive plates, competing tools couldn't do that part. Because like I said, Riffyn is all about getting structured data that's ready for whatever you want to do to a data frame, any kind of manipulation you want to do to a data frame. Riffyn can do this really well. Databases like those LIMS systems I was describing can't really handle this, and definitely unstructured data can't handle this either. So what I was able to do, I had a 6-week project with Imperial and a couple of other partners. And what I did was I built a really simple executable web app that lives on top of our Riffyn platform that's just specific for these labs. They don't want to have to learn a new software. They want something that's made specifically just for them. And so it's just a Flask app written in Python that's run on API calls. Riffyn's API is really robust. And so this is something else that you can do with our tool, is you can use it to make as many other tools as you can imagine. And so, yeah, that's kind of the end of that little case study. So that was maybe start to finish. I was in the UK for 6 weeks working with Imperial last winter. And so maybe start to finish that project was like 2.5 months. And that was just part of my job. So during that time, so when I get to work at Riffyn, I get to work on lots and lots of fascinating, yeah, really amazing science. And so that's my talk. Data is the new science at Riffyn. I hope it was helpful. Thank you for your attention.
Thank you so much for that.
Yeah, thank you so much. So it was really interesting to hear from a scientist company. We'll be taking questions at the end and hopefully have a little discussion going. For people who want to post any questions that you think during the talk, you can use Q&A area in the event tab. And you can also upvote questions that you find especially interesting. So we'll have Dr. Yang present next. We've hopefully fixed the screen sharing issue. So if you'd just like to share your screen, then yeah.
All right. I think this is working now, I believe.
Yeah, that works.
Technology, huh? I was joking that this is digital equivalent of when we're physically presenting, right? Every time, like we have problems connecting to the projectors, right? When a bunch of IT people are having trouble connecting to the projector. So turns out this is a permission issue that Mac has with the Chrome. You have to give Chrome specific permission to do the screen recording. So anyway, without further ado, so thanks a lot for Olivia and organizers to invite me to give this talk. So today I'd like to share with you sort of my experience and journey of how I get into the sort of software engineering business in synthetic biology industry, and what we actually do in Ginkgo regarding to the software. So I think I give a brief introduction of my journey, how I get into this. So back to my undergrads, I was actually majoring automation in Shanghai Jiao Tong University in China. So I was particularly interested in automation because I love how we can use robotics and use all sorts of interesting, cool stuff to make things easier. So I studied a lot of broad electrical engineering, computer science topics, and focus on doing the control theory, which is the automation part. So I went to graduate school, right? And actually, I was planning to do research in sort of this wireless communication stuff. There was some pretty interesting, cool ideas there. By bumping to this professor who were like doing this course called Intro to Synthetic Biology, right? And talking about how you do cell programming, right? How you can actually program living things. I was like, this could actually be the future, you know? When you think about it, right? You can right now we can program computers, right? Program electronics, right? But what if we can actually program living things, right? That could have huge implications and impact on the entire human species and the entire planet. That was something really got me to thinking, right? And I got really fascinated by synthetic biology and started actually getting really into it. I learned a lot of lab work, how to build like genetic circuit in cells. So at a part of my thesis, I actually built a bistable switch in yeast that sort of mimics the bistable switch in electronics. But in the meantime, I find this doing the lab work, right, is really sort of repetitive work. So it became really interesting to how you can use software, right, and use automation to build abstractions, right? It's like in the computer science where you you're always thinking about how I build abstractions, right? When you think about how we get into like first programming directly, like the sort of zeros and ones into the chips, right? Where now we're using high-level programming language like Python to actually like write things like to computers, like how we talk to people, right? It's pretty amazing. Can we do something equivalent like that in molecular biology, in this synthetic biology? So that's what got me really interested, right? So I ended up did a lot of soft work. The software piece I contribute a lot called Aquarium that can sort of model what your lab process is, then build abstractions so you can actually think about how you design experiments, then separate out the design and execution. So that lead me pretty naturally to Ginkgo, where Ginkgo is using a lot of software and automation, particularly robotic automation, to do the synthetic biology. So I started in Ginkgo in 2017 as a senior software engineer, and early this year, got promoted to principal software engineer. So a lot of work I do here is essentially in really similar spirit what I did in graduate school, to build that abstractions, right, between what you physically do in the labs and how you can take a level higher, right, where you think about how you actually program the cells, right? So like this is a quote from one of the Ginkgo's founders, Tom Knight, who's also a sometimes called the godfather of synthetic biology, who actually is the pioneer of starting this field. Says the interesting thing to program in the 21st century isn't going to be computers; it's actually biology. So if you think about so this is the Ginkgo's mission. Ginkgo's mission is to make biology easier to engineer. I think people who actually deal with the biology knows, right, how hard it is to actually engineer biology. So if you think about a lot of work that the academics do, right, that I spent like 4 years, right, building out a simple genetic switch in cells. It just like beyond your imagination. If you think about how we can program like electronics, right, it's supposed to be like really easy to do that, but in biology it's like messy and it's hard, and everything doesn't work as you expected the first time. You have to try again. So this is really Ginkgo's mission, is to make that process easier, right, so we can achieve great and big things. So I think the sort of basic concept, right, is that the DNA code is the common code that governs almost all the living species on Earth, right? And the point that now we actually have the capability to both write the DNA in terms of DNA synthesis, actually sticking to the cells, and also read DNA. So it's a lot of like we can program cells like how we program computers, right? You can actually enter the code into the computers and actually have a debugger actually tells you what the computer does. So that's sort of the basic premises of how the cell program business sort of based on. And if you look at the pandemic, right, what the COVID-19 pandemic sort of taught us is how sort of the biology, right, and cell programming is so critical to our lives, right? If you think about the COVID virus, right, it's just a piece of DNA code, RNA DNA code that actually can have huge impact to our whole society. Whereas we're actually using cell programming, for example, the mRNA vaccines, right? It's a piece of thing that we program that actually can be injected into cells to produce like spike proteins, right? Then can trigger immune response, right? And this mRNA vaccine is really amazing that you can actually program it, right? Meaning you can write the DNA code really fast. And for example, like it takes like a year to get this sort of from like from the start to actually deploy. It's like historical. And never in the human society we ever get to a vaccine so fast, right? So this is really the idea of synthetic biology and cell programming is really taking shape in real lives and really making huge impacts. So I I think sort of a broader picture, right? What really get me up in night is like this huge opportunity, right? Huge like impact they can make with cell programming, right? Sort of native, like in pharma, in biotech space, right? You have this vaccine production, vaccine manufacturing, like this is really already taking to shape, right? And a lot of therapeutic applications using gene therapy, using microbiome therapeutics are really taking shape because of cell programming, and also have tremendous impact on how we can shape the sort of environmental impact. For example, like Ginkgo is working, have a venture called Alonia, actually working out on the PFAS degradation with cell programming. So another field of food and agriculture, like think about the sort of we can design, for example, microbes, right, to actually do nitrogen fertilizers, right? So that will be a huge savings to the cost and also to the CO2 emissions to the world. And also like the consumer technology, a lot of materials that go into the consumer technology, including like sort of like a lot of things you use daily, and also things like perfumes, things like flavors is all like very impact, like being disrupted by cell programming. So what Ginkgo does in this field is we're building out a horizontal platform to do the cell programming across all industries. So we're not particularly tied into one specific industry. We're doing this, building this platform so we can actually be used across all different industries. So that means we partners with a lot of companies, Fortune 500 companies, but we also start sort of new ventures with other companies or with venture capitals to actually putting the applications out to the world. And on the technology side, Ginkgo is leveraging a lot of state-of-the-art technologies, including DNA sequencing, robotics, and even the state-of-the-art, the sort of the screening technology. For example, like the Berkeley Lights, that building this platform to really make the cell programming easier and scalable. So this is a sort of a simplified picture of what Ginkgo's sort of core capability does, right? We call it the foundry, and we're programming the cells using our foundry. So it goes to the sort of traditional sort of DBT cycle, right? You do design and you do build and you do test, and we also do fermentation in-house, where we can do both scale up and scale down fermentations, and do the process development and do the deployment. So we can do all this in-house, but the product actually commercialization is relied on our partners. But we do all the sort of R&D and scaling using our capabilities. And underpinning all these capabilities is our software and robotic automation platform that really allows us to scale dramatically year over year. So if you think about, if you look at the past about 6+ years of how Ginkgo's scale has grown, the sort of foundry scale is roughly scaling about 3x per year. And that's measured by like multiple metrics, right? By the number of unit lab operations, aka the number of liquid transfers we do on the robotic platform, the number of string tests we do that actually produce data on a specific instance of the string. It all grow pretty much exponentially 3 times per year. And some of the data that we have on our software, right? We have up to this month, we have about 5 million molecules being sort of built in Ginkgo, and 38 million samples, 25 million data sets, 61 million lab operations, 5 million like foundry service requests. So one question you will natively ask, like how do you handle all this large scale of things and data, right? that's like really lies on our software infrastructure. So this is an overview of a sort of simplified overview of our software application sort of system. So from the beginning part, sort of the design part, we have a DNA design tool. We call it Loom, that where the both the program teams and the internal design team can design large-scale combinatorial DNA designs, and we can send to both a external vendor, for example, Twist, or our internal DNA synthesis platform to do the DNA synthesis. So this is the part where you can actually write the code, right? Write the DNA code, right? And then once you have the DNA ready, it goes into our Foundry services. So we have a platform called Servicely. That's our UI and API for Ginkgo's Foundry services. So Ginkgo's Foundry services is basically the sort of the abstraction of our Foundry capabilities, right? For example, our NGS service, our high throughput screening service, our string construction service, right? All those DNA goes into this services, and eventually pumping out of that is sort of new strings, right? New data, like new insights into how your design works, right? So in terms of sort of a little bit go into lower level how these things got executed, right? Servicely interacts with our workflow design and execution platform called Organic. That's a software platform for all the workflows, including the liquid transfer and sort of bioinformatics updates, and that can be directly coupled with our robotics execution platform. When you have a liquid transfer, you need to be carried out a talk to the robotic execution platform to actually carry out those liquid transfer operations. So after all this sort of execution finishes, we have a data pipeline that deployed on Airflow that process the data in many different ways, and the end data got ingested into our data store, where that's our data management system. And underpinning all these activities is our LIMS system, stands for Laboratory Information Management System, that stores all our samples, containers, molecule strings, sort of things, both sort of virtual and physical, and that's underpinning all the applications. So a lot of sort of skill data I showed you earlier that's directly come from our LIMS. So to give you a sort of further example of our specific software applications, for example, one of the applications called Servicely, that's our UI and API for our Foundry services. That's essentially abstraction of Ginkgo's Foundry capabilities, right? Whereas the program teams, right? So sort of like back a little bit. So program teams is this teams in Ginkgo where they specialize to do the custom projects, right? They're individual teams, each responsible for a particular custom project. And what they do is they leverage the Foundry capabilities to carry out most of the work, right? So they themselves do sort of little of the work in terms of actually physical lab operations. They sort of outsource or insource all the work to Foundry, Ginkgo's Foundry, right? So what the platform they use to do that is using Servicely, right? They can request and get results, right, from the Foundry services using this really easy to use platform. On the other hand, the Foundry teams, right, can use this platform to manage all the requests and actually run the Foundry services. And on the other hand, there's a sort of developer for the services, because we have so many Foundry services and some are very complex to actually develop and manage. So we actually have a sort of equivalent of how you develop an app on sort of iOS App Store, Android App Store. Like we have an SDK that you can actually develop service on this platform. So the software side, in terms of how we build the applications, the front end, we use React, which is a popular JavaScript front-end framework. And sort of between the front end and back end, we have the GraphQL, which is really nice sort of abstraction how you present the data to the front end in native sort of JSON JavaScript format. On the back end, we are like sort of like majority of Python shop, right? We use Python and we use Django to build the backend of our applications. And we also use a Django REST framework to build the REST endpoints. And the database for this application, we use Postgres. And the application itself is deployed on AWS. So it's really easy to scale and pretty fairly easy to maintain. Another application example is Organic. I mentioned this are sort of a workflow design execution platform. It stands for Organism Construction Kit. It's an abstraction of both the laboratory unit operations, both physically and informatically, meaning that it have basically steps representing the liquid transfers, right, the physical liquid handling, but also the informatics, right, like doing the molecular manipulations, such as the PCR, or doing a digest, or doing a DNA annealing, right? We can do all those simulations, right, in the software, right, to actually match what you physically produce in the lab. And it's also very composable, meaning you can almost compose any type of steps together into a graph. This is our primary tech stack, right? On the sort of backend side, we mostly use Python and Django to do our backend. Front end, we use React and GraphQL. And on the sort of software, sort of more on the deployment side, we use Docker to do the containerization, and we deploy the Docker on a container orchestration platform called Rancher. And we use GitLab to basically track our code and do the CI/CD continuous deployment process. And also on the sort of data analytics side, we use Airflow to build those sort of DAGs, right? Graphs of data analytics, right? A lot of bioinformatics workflows are built that way. And all those tools are deployed on AWS. So it's very easy to scale to meet our ever-scaling needs. Last but not least is like, I know a lot of folks here are either sort of mostly in the software career or like in the still studying computer science, right? So the question is, why do you want to consider a software career in synthetic biology industry, right? Like natively, right? If like you think about it, you will go to the big tech companies, right? To do like Google, Amazon, right? This is like sort of do AI, right? Those things. But why choose a career in say biology, right? So this is a couple bullet points I really believe in, right? I think this is a synthetic biology is just started a huge technology wave that's really I think could be as big or even bigger than the beginning of computer industry or the internet. It could be really, really big as when you join at this time. Is still we're still far away from the promise. We're still starting, so it could be rewarded both intellectually and monetarily if it's successful, right? And there's so many challenging problems that software could be key to solve in this industry, and you can really make a difference here, make a bigger difference here. And another thing I think I really like is if you look at this article that our CEO Jason Kelly wrote, is that why do you innovate with biology? It's because you cannot eat software, right? You cannot dress software. A lot of things we ourselves are biology, right? Think about the COVID pandemic, right? How like biology can affect us in every different ways, and the ability to really program the cells, right? It could have huge impacts. And your ability to actually make software to do cell programming, it's just like I cannot think of a better way to do that, right? Like I have a link here for Motif Foodworks. That's a venture we actually started doing the using basically the synthetic biology to program cells to produce the ingredients that can make, for example, like similar to how Impossible Burger tastes, right? You produce this like it totally change how you can think about the future of food, right? another thing to consider is like, especially when you're young, I personally would recommend a field that still is like in its young phase and still has more potentials. You have much more fun and thrilling ride. Another important thing to remember is that you don't need a PhD in biology to join this industry. In fact, most of our software engineers in Ginkgo don't have a bio background to start with. So as long as you're open and passionate to learn about this, you'll be really successful here. If you want to learn more about software engineering and what we do in terms of digital technology side in Ginkgo, welcome to follow our tutor called Ginkgo Bits, because we're using bits to write software applications to programming cells, and we also have very active blogs tell you what we do, both in technology side, also sometimes could be very whimsical side. So follow us. That's it. So happy to take any questions later when all the talk finishes. Yep, that's it. Thank you.
Thank you, Dr. Yang. That was very interesting. As you heard, you can go and check out Ginkgo's blog and their social media. And yeah, so we'll now welcome the third speaker, Dr. Donovan from Healx. Dr. Donovan has experience in a range of things from web tech to bioinformatics and co-founded a wearable tech company before joining Healx as the technical lead in machine learning. So glad to have you, Daniel, and share your screen when you're ready.
Cool.
There you go.
Thank you, Olivia. Can you hear me? Just checking.
That's good.
Yep.
Great. Thank you. Great. So thanks, Yaoyu and Zoey. Some great talks there. It's amazing how many kind of themes I'm so familiar just from listening to some of the problems and things that you've described. So I'll just give a brief bio who I am. So I'm an AI tech. I am the AI tech lead at Healx. So I have a background in biochemistry. Before that, I was kind of doing maths and physics, and spent a while doing a few postdocs before switching to industry. So I've got 15 years of computational biochemistry type machine learning experience. I was one of the first employees at Healx, so I've got to enjoy working with the company from being, just a handful of people sitting around a table where everybody does UI, backend, and science all together to, a more specialist role. I helped research and implement some of our first prediction pipelines, which I'll explain later on in the talk. And I've also helped, well, as part of that, I've helped bring some promising treatments for some horrible rare diseases to almost to market. We're in phase 2, clinical phase 2 with some of them now, which is very exciting. So first, I'll explain a little bit about Healx and what we're working on. So we are looking to find treatments for rare diseases. So a rare disease is a disease that affects fewer than 1 in 2,000 people, often a lot less frequent than that. And rare diseases are often seen as satellites of more well understood diseases. For example, diabetes is often seen as a cluster of rare diseases, and you very rarely see the individual constituent disease clusters of diabetes by themselves. 8 out of 10 diseases are classed as rare, and a sheer unfortunate side effect of statistics is that around 1 in 17 of us will be affected by a rare disease before the end of our lives. These rare diseases are largely without treatment. 95% of them have certainly no known cures and very few known treatments. There are around 400 million patients suffering with rare disease worldwide, and unfortunately a significant number of those are also children. So at Healx, we have a simple mission. We are using AI to identify and progress novel rare disease treatments at scale. We have a long-running mission statement that every rare disease patient deserves a treatment, and it's certainly something that's it's been with us from the start. So how do we use AI to solve these rare disease challenges? Typically, rare diseases, by virtue of being rare, are not very well understood. So we need to use NLP to complete gaps in the knowledge of these rare diseases. As I said, 95% of these diseases have no treatments, so we're using machine learning to find existing treatments, repurposing drugs for these rare diseases. And also, by virtue of the small market size for these rare diseases, they don't make traditional targets for big pharmaceutical companies. So oftentimes there won't be treatments simply because there won't be a return on investment for bigger companies using traditional drug discovery pipelines to find this. So we've demonstrated that we can use AI to automate a lot of the processes, shortcut a lot of the expensive investigation stages, and bring treatments to trial and hopefully patients much sooner. So our chairman, Dave, is better known as the co-inventor of Viagra, but he's a repurposing specialist. So he's the best way of discovering a new drug is to start with a known drug. So at Healx, we do a lot of drug repurposing. Sorry, kids are going to bed in the background here. So a repurposed drug is a drug which is already on the market for another treatment or indication. And the idea is that if we can find an alternative purpose, an alternative treatment for this compound, we already have a lot of the kind of initial clinical trial data to show that these drugs are safe to take, and we can shortcut a lot of the a lot of the expensive stages. So this is a comparison of the traditional drug discovery process with our AI-powered redevelopment and discovery. Typically, the traditional process is a lot slower, can take up to 14, 15 years, and there's a much lower probability of success. You know, you could fail. The compound could fail at any one of these stages. By having fewer stages, shorter process, and maybe a more intelligent way of selecting our predictions, we've demonstrated that we can bring products to patients in a shorter time and with a higher probability of success. So I think I imagine the bit that most people are interested in is the technology that we use at Healx. Certainly what I'm working on is super interesting, I find. So we have a platform that we've termed HealNet. We call it our knowledge graph. It's a typical data graph, but with a very specific biomedical use case and consequently specific data in there. So we take a lot of data from public sources, and we also purchase data sets with, use this as a high quality scaffold. So data sets like Drug Bank or Kemble or OMIM and sources like this. We have a team of in-house curators who are rare disease experts or pharmacology experts, and they provide additional curated information either on the rare diseases we're interested in or compounds that we're interested in, just to kind of kickstart predictions. And we have this NLP source, which is very expansive. We mine sources like PubMed and other places to kind of pull out the state-of-the-art understanding of the biomedical world and merge this, assemble this into our graph. Once we've built the graph, we can start doing reasoning and prediction. So we have a battery of methods that are all optimized for predicting different aspects of drug predictions. Once we've produced an in silico list of predictions, we do pass the results onto a team of pharmacologists and drug discovery experts who are really kind of the final gate in producing our predictions. They will go through our lists and tease out the predictions that they find the most promising. And as a result, we produce combination therapies and also monotherapies for the rare diseases. Some of the techniques that we use here, I'll just go over them quickly. There's traditional gene expression matching. So this is quite similar to the CMAP method, which some people may be familiar with. And then we have some more typical machine learning type problems. This method 2 is a similarity matching method. So we create a disease space built of similarities between diseases, so phenotypic similarities, genotype similarities, other information that we have, and a space of compounds, so compound structure similarity, I guess compound target similarity, even kind of like ATC similarities. And we build this high multi-dimensional space. We have known compound treats relations, and we use simple tools like support vector machines to kind of slice down this compound space and find which of these compounds are likely to treat disease. We have some knowledge graph methods. So in our graph, you can draw paths between drugs and diseases, and some of these paths are we train models on these paths, and we can show that some of them are more likely to be curative paths. So then we have predictions, but predictions with hypotheses behind them, I guess. Some more methods. So there's another knowledge graph method, a completion method, which is similar to Notavec type problems, if people know that from the machine learning world. Some of these are deep learning type methods, more drug gene expression matching and pathway matching. So this is where we're at with some of the diseases we're looking at working on at the moment. Typically, we have a kind of a cross-functional matrix team organized around whatever disease we're working on. That team consists of curative experts in the biology of these diseases, pharmacologists, bioinformaticians, machine learning specialists, software engineers, DevOps people, and they will spend 2 to 3 months just focusing on this disease, making sure that we've got all the data we need, running our predictions pipelines and producing lists of predictions, which we can then take into preclinical validation. So sending these compounds off to labs, seeing if there's any effects. And so far, we've had quite promising results, and we've been able to take at least 2 diseases into clinical trials, and we're hoping to bring our 3rd and 4th in very soon. This is us really. So we've recently, in the last year, we've gone from 40 employees to 80 employees, and we're looking to scale. Again, by next year, we're going to hopefully double in size to around 150, 160 people. So we're actively hiring. We're on the lookout for all sorts of driven and passionate people. I imagine folk in this forum are going to be synthetic biology computational experts. We love bioinformaticians, and we want more of them. So please to look at our job openings. And alternatively, if you can't find a role that doesn't quite suit your expectations, but is if you'd still like to apply, then we welcome speculative applications as well. That's yeah, it's great fun. Thank you. Olivia, I'll hand back to you if I can.
Fantastic. Yeah, thank you so much. That was another incredible talk. And I think we're ready to move on to some questions. So in the stage tab in the Q&A, if you still have some questions that you want to throw out, feel free to do so. Otherwise, we'll start with the most upvoted question, which is not test by Anonymous, but a question for Dr. Zoey Herm. If you would maybe like to reshare your there you go. So how did you get the computer skills in order to work at Riffyn as a software developer?
Yeah, I saw that question. So I think I could have clarified slightly in my talk. So I'm not a software developer. I am an application scientist. So I work with customers with the scientists at the customers who buy our software, and I like learn all about what the science is that they're doing, and kind of guide them and collaborate with them to get their work into our platform. And so I'm not doing the software, like any of the back end work at all. I do some coding, definitely not, I don't want to even say programming. I do like some coding with our API, and I learned those skills. A lot of it was self-taught. I decided that I want to, between my last job and working at Riffyn, I decided I wanted to pivot pretty heavily into data type work because it was just where I found the most joy in science. So I went to one of these data science boot camps, but I left after 6 weeks because I got this Riffyn job halfway through. And so I was, left to actually do the data science instead of learning about it. So that's where I learned those things.
Thank you. So this is a question kind of everyone. Open source are good if you're a software engineer in synthetic biology.
Yeah, I can take that question. So in Ginkgo, we actually open source quite a couple projects we actually use in Ginkgo. We find could be useful for public, so we actually open source it. So if you go to our GitHub page, you can actually find all the projects we open sourced. I will recommend one I personally sort of invested a lot of efforts in. So we have a genome tracking tool called Edge. That's really actually excellent in terms of tracking genome changes, right? Basically, when you're doing synthetic biology, right, you do a lot of genome modifications, right? You insert like a piece of DNA into the genome, right? And then you keep modifying that, right? Generation over generation. And Edge is a tool to efficiently track those changes over many multiple generations, right? It's a Python sort of tool, Django. So recommend to check it out if you're interested in contributing open source. There's also other various open source projects out there. I know, like in the bioinformatics field particularly, there's a lot of open source projects that really need people to contribute. So.
Or any tips for people that are software engineers and just want to get involved in some way? In that case, let's move on to the next question. So I guess this is one for Dan. Which computational skills do you look for at Healx? Would it be better to learn more about building machine learning models or learn about how to extract novel insights from data instead?
Good question. So I mean, we're interested in both, really. So I think it depends which role you're looking for. We have more science kind of based roles in the machine learning and the bioinformatics side. So our scientists typically, their goals are to publish papers, to open source their methods that they're working on, and to kind of extend the knowledge of drug repurposing, drug compound treatment prediction. So certainly, well, I guess both learning models and extracting novel insights there. But on the other side, we hire a lot of engineers, and what we look for engineers, maybe not necessarily be an interest in publishing, but kind of an excitement in the field. When we interview people, we have a brief kind of face-to-face chat and technical screen, just a coding session, live coding session, and you really get a feel for just the conversation with people. If people are actively enjoying what they're doing, or if they're some people are not quite enjoying it quite so much. And we certainly found that the people who are passionate about what they do, they understand the tools they use, and they've read around the latest kind of ideas and developments, and they are certainly the sort of people that we're hoping to employ. I've probably gone off track. I'm not sure if that was quite the answer.
There is another question for you that's quite closely related. It's in the chat channel, actually. So which tools are you using for your data analysis and management for all of these pipelines? And also, would you mind sharing what percentage of budget is spent on IT for a new age company like Healx? And would you like to use Ginkgo's platform?
Yeah, good, again, good questions. So which one first? The pipeline tools we use are open source pipelines usually. It's very much the best tool for the job. We try to be as agnostic as possible. So if something doesn't work, then hopefully we're not too tightly coupled to anything, and we could just switch from one to the other. At the moment, we are using Airflow, like Dr. Yang, quite a lot. It's a good tool. But what percent of our IT budget? So this is where it gets interesting, because we're, as a traditional IT company, as a biotech company, we a lot of our budget is going to be spent on running models and clinical trials. If you look at the cost of running a clinical trial versus running a tech department, it's nothing really. So we find that because everybody's looking at comparisons, we actually have quite a lot of leeway with our budgets for these computational sources. Maybe I'm not sure if I should say this, but no one so far has told me off for spending too much on cloud services. It's not unlimited budget, but it's certainly never going to be the scale of clinical trials. So it's coming from academia and coming from a very small startup where you have to watch every penny to being able to, if you want to train BERT on a 16 GPU machine, then you do it. You don't have to kind of fill in any forms or kind of reserve an instance weeks in advance. It's very liberating. Would I like to use Ginkgo's platform? Well, of course. Actually, I think maybe also Riffyn. There's some we get through a lot of notebooks. I've got my notebook here, and a lot of the things I'm writing down are really not very well suited to pencil and paper. That sounds like it would serve a lot of problems for us. Yeah. Shall I hand back, Olivia?
Lovely, thank you. So we have another question in the chat, which is how can non-technicals get involved in the industry, and what kind of routes do they go in? What is the product scene in general in biotech and software like? This was from Jack Chong.
So I can try to answer that question first. So on Ginkgo side, we actually have a software product management team, about like 8 to 10 people close. So they're sort of more on the, if you call it non-technical, they're not writing software code, right? But they are sort of the visionary and the bridge for our software or our software customers, right? They do a lot of user studies, right? They figure out what the user needs, and they plan out the roadmap and vision for how the software should be, right? Should be built, what the features should be built, what the software applications we should build, what things we should prioritize. So if you're not really into sort of coding but really into this industry, I think the sort of the product management side is a good sort of way to get into this industry. So there's also sort of other things that are, I think, pretty interesting. Is if you're interested in the sort of commercialization of the technology, in particular the SynBIC, there's a lot of opportunities there, right? Think about what the wildest thing, like cool things or good things, of course, you can use the cell programming to do, right? Think about like how you can, I think we have project that like how you can program like your skin microbiomes, right, to actually be like source of the mosquito repellent, right? So like there's a lot of very cool ideas that you can push forward or on the commercial side. So that's another route I would recommend. Yeah,
and even kind of simpler on the commercial side is sales. Like the sales team at Riffyn is just a really special, talented group of people. So there's pre-sales, there's customer engagement. There are a lot of different ways, I think, to get involved. Like one person on there's an associate director of education at Riffyn, and she writes our help article documentation and like generates training courses and things like that. And there's people like me, like my role, who I'm, not interdisciplinary, if you want to think about it that way. So, and at Riffyn, we're only a 50-person company, and yeah, there's a huge product team. There's my team, like application scientists, there's education, there's sales. And so when you get to bigger companies, you can even further specialize. So I think there are a lot of different ways to get involved.
Thank you. So we have another question about the sort of relating to the skills that people need to get trained in preparation for things that use in industry. So from Noah Sprint, we have, do we need these new approaches to biology to move upstream to academia, where most biologists are still trained, to make the transition easier? Or are the use cases different / is the transition okay?
I have big feelings about that, and so there's a pause for them. Yeah, I mean, I do, having spent a decade plus in academia, I do think that there's a lot that academia can do, that advisors and faculty can do to prepare students for industry better, because there are many more jobs. The odds that you will stay in academia are very low. But that said, when you're like really in academia, you're learning critical thinking. And so if you really kind of make that your focus, to like be curious, to be persistent, to just be a good worker and critical thinker, those are the most important skills. So yeah, there's definitely bridging that could happen. Yeah, there's definitely bridging that can happen, but it's not like it's necessary for you to do things that are industry specific when you're in academia to be successful, I think.
Yeah, I would personally advocate for more programming training, or at least try get your hands dirty on like build stuff, right? Like I think complementary to sort of a traditional biologist training, right? I think have the really strong analytical skills, right? Using sort of Python, R, right, to do the data analysis could really make you stand out in your sort of, if you want to transition industry. Yeah. We personally, I mean, Ginkgo really value sort of biologists who have programming experience skill, right? Because you can do like you can leverage that skill to do a lot of things more efficiently with a higher throughput.
Oh yes, so we have a question from Alexis Custis. When can we expect full lab abstraction to do SynBIC? This way, infrastructure, the servers, etc. When can I expect to not care about labware at all?
I love to take that question, actually. This is like on my mind for many, many years, right? Since I started as a graduate student. I do believe in the future, at some point, we'll achieve that. That's sort of the grand goal, right? Of sort of my dream is to have, imagine a world where you can just like on your computer and have all the lab capabilities at your hands, right? Without you ever touching any lab equipment for a single second, right? Imagine that, like how you can deploy application on AWS, right? So I think eventually we'll get there, right? But I think the route to get there will still be long and very challenging. The reason being that like if you look at the sort of the variety of things we do using our infrastructure in the lab, it's just mind-boggling, right? And how particular also like how you can standardize those infrastructure is also very challenging. And I think it will take a long way to get us there. But there's already pioneers do that, right? For example, if you look at Stratios, right? They're having this cloud robotic labs out there, right? You can actually program that, right, without actually going to their labs, right? But in terms of achieving that full dream, right? I think we need a lot of more people joining this effort, joining this mission to make that happen, right? I think that will be really, really valuable in terms of advancing our society, right? There's so many challenges we need to solve, right? Climate change, health medicine that can be really step up by our capabilities to program cells.
Yeah, there's actually one Bay Area startup who is doing that. They're called Emerald Cloud Lab as well. And yeah, but similar idea.
Yep.
Definitely still in the vision stage.
Yep, definitely.
I was just going to add that, you know, we sometimes I think you can be limited by our imagination when approaching these kinds of problems. I spent 3 or 4 years, you know, no, actually longer than that, doing protein structure determination with NMR and X-ray and electron microscopy. And, you know, we always joked that, oh, one day somebody will just find a way to go from, you know, protein sequences to thirds, you know, to structures. And, you know, but we didn't believe it. And now, you know, last year someone did. And it's, you know, it's amazing what you can do. So I just don't, I mean, we might not be there yet. And certainly with drug trials and things like that, I think people would be very reluctant to take any kind of medication if it hadn't been, you know, properly validated in laboratories first. But, you know, but to kind of stick to that way of thinking is, you know, we're never going to get anywhere. So yeah, just, you know, be imaginative and keep pushing the boundaries. I think it's, yeah, very important.
So much. We're just about running up to our. We could potentially take one more question if anyone would like to throw one out. Otherwise, I have a question. So, having worked with some synthetic biology data standards like SBOL and the accompanying Python languages, how do you think that the data storage and the kinds of data that people use in all of this space has to kind of evolve along with the software and the automation that are being used to manipulate it and to get insights from it and build things out of it? This is to everyone as well.
So I think Zoe is probably more qualified to speak on this than I'm not familiar with that data format, but I mean, everywhere I've worked, we've had some unusual binary data format that's been produced by a microscope that is now out of manufacture, and nearly all the time we can get the data back out again. It's just a bit of a pain. So as far as standards go, open standards would save everybody a lot of time. If you can store it as a CSV file, store it as a CSV file, and it'll make everybody's life a lot easier. That might not be in your answer, though. I'll hand over to Zoe.
Yeah, sorry, I think you were cutting out. I was going to defer to the two of you because I think you were cutting out a little bit, Olivia, so I didn't totally hear your question. I thought it was about something different. But from Dan, from your response, what I could glean, Olivia, is that you were asking about proprietary data formatting in scientific instrumentation and that being problematic. And if,
Yeah, I can repeat it a bit. I guess that would be part of it as well, and kind of just how do the data standards that we use have to change and evolve to accommodate for the growing the needs that are coming onto the horizon.
Got it. Yeah. There's this data standard actually. It makes a lot of sense. I have to look up exactly what it stands for. But I'd never heard of it before I moved to Riffyn, but it's called FAIR data. And that stands for, I don't want to get it wrong, findable, accessible, interoperable, and reusable. So I guess everyone is aware of this. So I think that the reusable portion of that then goes to this, goes to what Dan was saying, that, like you need to have things in CSVs, like maybe XML, like you have to, but like just put everything in a CSV. And yeah, I think that's the right thing to do. I think that I think that customers of people buying instruments or people buying tools are starting to push back. There used to not be a lot of power, like consumer power. Like if you have like a big company like Thermo Fisher Scientific, and they're generating, their whatever platform that they're using, if the data that you get can only be, manipulated using that platform, people just didn't really have a lot of other choices. But I don't know, maybe with new venture-backed companies being able to get into the market when they're smaller, this kind of thing. It does feel like that tide is shifting a little bit. So hopefully that starts to talk about your question.
Thank you so much. Would you like to add something, Dr. Yang?
Oh, I'm good. I'm saying that there's a question for Zoe in the chat, particularly about Riffyn.
Yes. So yeah, Zoe, how much time does it take to learn/train to be able to use Riffyn, and how much does Riffyn cost for small teams?
So like I said, I work with a very awesome sales team. So I encourage you to go to our website and reach out to them to talk about pricing information. But learning how to use the tool, I can train people. So there's kind of like two classes of being able to use it. One of them is just capturing data in it. And so like someone has already made a workflow for you and you are just going to execute on that specific workflow. I can train people on that in like 90 minutes, 2 hours, something like that. And then you do a couple of hours of practice on your own. And then you're pretty much ready to go for capturing live data in the lab. But if you want to build those templates, that's going to take a little bit more time and a little bit more practice. We have a lot of, in terms of how much time, it depends on if you want like live teaching sessions from someone like me. I do a lot of training, or if you want to do it totally self-paced with some of our online tools. So not a day, but also not a week, I guess, something in there. I don't know, would be an estimate. But yeah, I encourage you to just go to riffyn.com, and then there's like a bunch of different places where you can reach out to our team if you are curious about pricing. But for small teams, I think it's affordable.
Thanks so much. We have pretty much run out of time. I don't want to keep our speakers any longer. And I'd like to thank you all again for joining us today at Imperial College and sharing your perspectives with us. It's really great to hear from such different areas as well. in a very similar industry, there's still so much variety and innovation going on. So that's for students. Yeah, thank you so much. And I hope to see our audience again at our next SynBIC talk, which will be announced. We will also share the YouTube link when it is ready in the next couple of weeks.
Sounds
Thank
good.
you,
Thank you,
Olivia.
Olivia.
Thank you.
Thank you. Thanks, everyone.
Everyone.
Right.
Yeah, thanks, everyone.
Thank you. Have a good evening. Bye-bye.
Bye-bye.